Sexual reproduction in flowering plants. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfume that we swoon over, the rich color that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own purpose, our selfishness. All flowering plants show sexual reproduction. A look at the diversity of structure of the inflorescence, flower, and floral parts shows an amazing range of adaptation to ensure formation of the end product of sexual reproduction, the fruit and seed. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plants, that is, angiosperms. Flower, a fascinating organ of angiosperm. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the name of 5 more flowers that are used in social and cultural celebration in your family. Have you heard of horticulture? What does it refer to? To a biologist, flowers are morphological and embryological marvel and site of sexual reproduction. In class 11, you have read the various parts of flower. Figure 2.1 with the help of you recall the part of a typical flower. Can you name the two parts in a flower in which the two most important units of sexual reproduction develop? Pre-fertilization, structure and event. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and the female reproductive structure, the androecium and the gynoecium differentiate and develop. You would recollect that the androecium consists of a whorl of stamen representing the male reproductive organ and the gynoecium represent the female reproductive organ. Stamen, microsporangium and pollen grain. Figure shows two parts of a typical stamen, a long and slender stalk-like structure called filament and the terminal generally bilobed structure called anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and the length of stamen are variable in flowers of different species. If you were to collect a stamen each from 10 flowers and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under the dissenting microscope and making neat diagram would elucidate the range in shape and attachment of anther in different flowers. A typical angiosperm anther is bilobed with each lobe having two theca, that is, they are diathecous. Often a longitudinal groove run lengthwise separating the theca. Let us understand the various types of tissue and their organization in the transverse section of an anther. The bilobed nature of anther is very distinct and the transverse section of the anther. The anther is four-sided tetragonal structure consists of four microsporangia located at the corner, two in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grain. Structure of microsporangium. In transverse section, a typical microsporangium appears near outer circular in outline. It is generally surrounded by four wall layers: the epidermis, endothecium, middle layer, and the tapetum. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grain. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how a typical cell could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupy the center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrad. What would be the pleiade of the cells of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspore from a pollen mother cell through meiosis is called microsporogenesis. The microspore, as they are formed, are arranged in a cluster of four cells, the microspore tetrad. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grain. Inside each microsporangium, several thousand of microspore or pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grain represent male gametophyte. If you touch the opened anther of a hibiscus or any other flower, you would find deposition of yellowish powdery pollen grain on your finger. Sprinkle these grain on a drop of water taken on a glass slide and observe under a microscope. You will be really amazed at the variety of architecture, size, shape, color, design seen on the pollen grain from the species. Pollen grain are generally spherical, measuring about 25 to 50 micrometer in diameter. It has a prominent two-layered wall. The hard outer layer, called exine, is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand with high temperature and strong acid and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exine has prominent aperture called germ pore where sporopollenin is absent. Pollen grain are well preserved as fossil because of the presence of sporopollenin. The exine exhibit a fantastic array of patterns and designs. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. The vegetative cell is bigger, has abundant food reserve, and a large irregularly shaped nucleus. The generative cell is small and float in the cytoplasm of vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperm, pollen grain are shed at this two-celled stage. In the remaining species, the generative cell divide mitotically to give rise to two male gametes before pollen grain are shed. That is three-celled stage. Pollen grain of many species cause severe allergy and bronchial affliction in some people, often leading to chronic respiratory disorders like asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came from India as the continental with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Pollen grains are rich in nutrients It has become fashion in recent years to use pollen tablet as food supplement. In Western countries, a large number of pollen products in the form of tablet and syrup are available in the market. Pollen consumption has claimed to increase the performance of athletes and race horses. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grain retain viability? The period for which pollen grain remain viable is highly variable and to some extent depend on the prevailing temperature and humidity. In some cereals such as rice, wheat, pollen grain lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae, and Solanacea they maintain viability for months. You may have heard of storing semen sperms for many animals, including humans, for artificial insemination. It is possible to store pollen grain for a large number of species for years in liquid nitrogen, minus 196 degree Kelvin Celsius. Sorry. Such stored pollen can be used as pollen banks, similar to sand banks in crop breeding programs. The pistil, megasporangium, ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one, the pistil may be fused together, syncarpous, or may be free, apocarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grain. The style is the elongated, slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, locule. The placenta is located inside the ovarian cavity. Recall the definition and type of placentation that you have studied in class 11th. Arising from the megasporangia, commonly called ovule. The number of ovules in an ovary may be 1, wheat, paddy, mango, too many, papaya, watermelon, orchid. The megasporangium. Let us familiar ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stall called funicle. The body of the ovule fuses with the funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integument. Integument encircle the nucleus except the tip where a small opening called micropyle is organized. Opposite the micropyle end, the chalazal representing the basal part of the ovule. Enclosed within the integument is a mass of cell called the nucellus. The cells of the nucellus have abundant reserve food material. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropyle region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. In MMC undergoes meiotic division. What is the importance of MMC undergoing meiosis? Meiosis results in the production of 4 megaspore, female gametophyte. In the majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develop into female gametophyte, embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. What will be the pleiady of the cell of the nucleus? MMC, the functional megaspore and female gametophyte. Let us study formation of embryo sac in a little more detail. The nucleus and the nucleus of the functional megaspore divide mitotically to form two nuclei which move to opposite poles, forming the two nucleate embryo sac. Two more subsequent mitotic nuclear divisions result in the formation of 4 nucleate and later 8 nucleate stages of embryo sac. It is of interest to note that these mitotic divisions are strictly filinuclear. That is, nuclear division are not followed immediately by cell wall formation. After 8 nucleate stage, cell wall are laid down, leading to organization of the typical female gametophyte or embryo sac. Observe the distribution of cell inside the embryo sac. 6 of the 8 nuclei are surrounded by a cell wall and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus of the large central cell. There is a characteristic distribution of the cell within the embryo sac. 3 cells are grouped together at the micropyle end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergid and 1 egg cell. The synergid have special cellular thickening at the micropyle tip called filiform apparatus, which play an important role in guiding the pollen tube into the synergid. 3 cells at the zalazal end and are the antipodal. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity, though 8 nucleate, is 7-celled. Pollination. In the preceding section, you have learned that the male and female gamete in flowering plant are produced in the pollen grain and embryo sac, respectively. As both types of gamete are not motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grain to the stigma of a pistil is termed pollination. Flowering plants have evolved an amusing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kind of pollination. Depending upon the source of pollen, pollination can be divided into 3 types. Autogamy. First, in this type, pollination is achieved within the same flower. Transfer pollen grain from anther to stigma of a same flower. In a normal flower, which open and expose the anther and the stigma, complete autogamy is rather rare. Autogamy in such flower require synchrony in pollen release and stigma respectively, and also the anther and the stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, viola, common pansy, oxalis, oxalis, and commelia, commelina, produce 2 types of flower: chasmogamous flower, which are similar to flower of other species which expose anther and stigma, and clistogamous flower, which do not open at all. In such flower, the anther and stigma lie close to each other. When anther dehisces in the flower buds, the pollen grain come in contact with stigma to effect pollination. The clistogamous flowers are invariably autogamous, as there is no chance of cross-pollination landing on the stigma. Clistogamous flowers produce assured seed set even in the absence of pollinator. Do you think that clistogamy is advantageous or disadvantageous to plant? Why? Genitogamy. Second, transfer of pollen grain from anther to stigma of another flower of same plant. Although genitogamy is functionally cross-pollination involves a pollinating agent, genetically it is similar to autogamy since the pollen grain come from the same plant. Now, xenogamy. Third, transfer of pollen grain from anther to stigma of a different plant. This is only one type where pollination occur during pollination bring genetically different type of pollen grain to the stigma. Agent of pollination. Plant uses two abiotic and one biotic agent to achieve pollination. Majority of plant use biotic agent for pollination. Only a small portion of plant use abiotic agent. Pollen grain coming in contact with stigma is a chance factor in both wind and water pollination. To compensate for this uncertainty and associated loss of pollen grain, the flower produces enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common among abiotic pollination. Wind pollination also requires that the pollen grain are light and non-sticky so that they can be transported in wind current. They often possess well exposed stamen so that the pollen are easily dispersed into wind current, and large often feathery stigma to easily trap airborne pollen grain. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The tassels you see are nothing but the stigma and style which wave in the wind to trap pollen grain. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. As against this, you would recall that water is a regular mode of transport for the male gamete among the lower plant groups such as algae, bryophyte and pteridophyte. It is believed, particularly for some bryophyte and pteridophyte, that their distribution is limited because of the need of water for the transport of male gamete and fertilization. Some examples of water pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine sea grasses such as Zoostera. Not all aquatic plants use water for pollination. In majority of aquatic plants such as water hyacinth, hyacinth, and water lily, the flowers emerge above the level of water and are pollinated by insect or wind as in most of the land plants. In Vallisneria, Vallisneria, the female flower reach the surface of water by long stalk and the male flower or pollen grain are released onto the surface of water. They are carried passively by water currents. Some of them eventually reach the female flower and the stigma. In other group of water pollinated plants such as sea grasses, female flower remains submerged in water and pollen grain are released inside the water. Pollen grain in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species,